2 tell us? Okay, what did we learn now from clue set 2? So what we've learned now from clue set 2 is that DNA is double-stranded instead of being triple-stranded like it was hypothesized in the past. They might not know this, but you might want to tell them that there were several hypotheses as to what the structure of DNA actually was. Is it a single helix? Is it a single strand? A triple strand? And so from the picture, the X-ray crystallography picture that Rosalind Franklin took, that let us know that A, DNA is double helix, and that DNA is twisted. It's a helical structure. It's twisted. But it doesn't tell us anything about where the bases are. It does tell us that the backbone, okay, the sides of the DNA are made up of the phosphates and the sugars, but it doesn't tell us exactly where the bases are. Are they on the inside? Are they on the outside? Okay. So now we're ready to look at clue set 3. So you pass out clue set 3 to them, and again, give them about 10 minutes to look at it, and then arrange, alter their models, and then go again. So what does clue set 3 tell us? And clue set 3 is Irwin-Chargaff's data, which tells us about the complementary base pairing, that A goes to T and G goes to C. So also in clue set 3, it also talks about how Watson made cardboard cutouts of the nucleotides, and he was trying to put them together to see how they might fit, because he was too lazy to actually read up on the chemistry of how they might bond together. He was just trying to how do they fit? And so based off of him kind of tinkering around with these cardboard cutouts, and finally looking at the data that Irwin-Chargaff presented, that let him know, oh, the bases are on the inside and they pair together A to T, G to C. So they need to, put their models together. And then to wrap it all up, you say, okay, so when you look at your models, how did you get them to fit together based upon the information in the clue sets? Okay. If they turn the models one side upside down so you can't see the print on them, tell them, oh, you need to take that apart because you need to be able to see all of the printing on all of the models. And so how do you get that together in order to do that? And what they should see is that they have to turn the other ones upside down, okay? Because, and so you ask them, so what do you observe about your model there? And they should say, oh, one side, okay, one side goes, one side goes one way, and then we need to turn the other side around. And then you can go, right, DNA is what we call anti-parallel. So I'm going to erase a little bit here. So if we have the DNA, I'm just going to kind of draw it like this. Okay, so from Chargaff's data, we now know that what does the adenine bond with? Okay, it bonds with a thymine. Okay, but I'm drawing these upside down here. Okay. All right. So we know that from Chargaff's data that this is how the bases pair together based on the chemical nature of the molecules. And you should notice that one side of the DNA runs one way, so let's say this one runs this way, and then the other side runs the opposite direction, okay? Because DNA is what we call anti-parallel. Kind of like a two-lane highway. So one side runs this way, the other side runs this way, okay? And then there are these bonds here that hold the bases together. Those are weak hydrogen bonds. Okay. And the bonds of the backbone are very strong, okay, very strong. And so Watson and Crick, they were able to then, based on all of the information that they read and from these other scientists' discoveries that were presented to you in these different clue sets, they were able to put that together and come up with the structure of DNA They wrote it up in a paper, a really short paper, not very long at all, published it, and then it was they were awarded the Nobel Prize in 62, I think, 1962. Now, they also made an observation based on the model of DNA that they put together. They made an observation about the nature of how DNA might replicate itself. They just proposed a hypothesis as to how DNA might replicate itself, and then other scientists worked that out later. And so just based on looking at the model here, okay, your basic model in front of you, can you propose a mechanism as to how DNA might replicate itself based on what you know here, okay? And so hopefully they'll get the idea. You might just have to kind of guide them through it, that the DNA can open up, okay, and so it can open up that the DNA can open up, okay, so it opens up, and then one side can be used as the template to build the other side, and the other side of the original DNA can be used to build the other side of the DNA for replication. So when it replicates, you're actually going to have DNA that is half of the original DNA and half of the newly synthesized DNA. So in blue here, I'm representing the original DNA strand, and then in pink, I'm representing the newly formed DNA. And what does that come out to? That comes out to 2 identical strands of DNA. 2 that's been replicated, and it's been replicated in this process. So again, the purpose of this lab was to look at the structure of DNA, yes, look at the structure of DNA, but to really understand that these discoveries aren't made overnight, and that scientists do collaborate, and new discoveries are credited to discoveries from the past as to how, we look at the historical, we look at how DNA was discovered. Not just that, oh, Watson and Crick did it, but look at how did they figure that out? They had to take the knowledge from all of these other scientists in history and put all of that information together in order to figure out the structure of DNA and how DNA transmits the genetic code, and then how DNA is replicated. Okay? All right, any questions? Okay. So again, the students might just want to jump right into those puzzle pieces. That's why I suggest you don't give them the puzzle pieces until they've had about 10 minutes of reading clue set 1. Because if you give them the puzzle pieces and clue set 1 at the same time, they're just going to want to play with the puzzle pieces. Okay? So I just forgot to get the puzzle pieces out for the thing today. But they're just paper, little colored paper cutouts of nucleotides that kind of fit together.
So how about clue set 3?
Clue set 3?
Is this the clue set 3?
Yeah, that's clue set 3, the Chargas data.
The second
Oh, okay.
one's right here.
Clue set 2, I just haven't, I want to laminate the picture, the bigger picture of the Rosalind Franklin's picture. And then I will, you'll have the folders up here. There's 12 clue set 1s, 12 clue set 2s.